Hello, amazing biology students. Today we are going to go over dihybrid crosses, but before beginning dihybrid crosses, I want to clarify a detail using a monohybrid cross and go over the purpose of why the heck we're doing this in the first place. Now, what is the point of learning this? The purpose of completing Punnett squares and genetic crosses allows us to make predictions about future generations, which can be important for decision making when breeding animals for food supplies or seeds for future crops, or even understanding how to prepare your own home for the possibility of having a child with potential genetic disorders. Now, I want to clarify a detail about what these letters represent using a monohybrid cross as an example. The two-letter genotype combinations, capital Y, and lowercase y, represent the parents. However, I want to make sure that it is clear to you that the letters at the top and the sides of the Punnett squares represent the gametes for each parent. This is a possible sperm cell that forms during meiosis, and this is the other possible sperm cell that forms during meiosis for this particular parent. This letter represents a possible egg cell, and this letter represents the other possible egg cell for this possible parent. Now, these gametes combine in each of these boxes to represent potential or possible offspring for the cross between these parents. Here's a possible child that formed when this sperm fertilized this egg cell. Here's another possible child, and here. I wanted to clarify that the letters above and on the sides of the Punnett square represent and what they represent before we move on to the dihybrid cross. Now, it is important for me to state that the dihybrid crosses are crosses that involve two different traits. These two traits are on different chromosomes. They're not linked together on the same chromosome. Therefore, they are capable of independently assorting during gamete formation. Here is a sample problem that we are going to use to go over how to solve dihybrid crosses in 4 easy steps. Step 1, identifying traits. Identify the 2 traits that are in the problem and write them out for future reference. For example, in mice, the ability to run normally is a dominant trait. I'm going to go ahead and underline this information. The recessive trait causes mice to run in circles only. Mice with this recessive trait that run in circles are called waltzing mice. I am going to underline this information as well and write down the phenotypes and list the genotypes for this trait. I'm going to use the word run to represent the phenotype for the normal running mouse. And because this is the dominant trait, capital R is one genotype possibility for the normal running mice, or capital R, lowercase r is the other genotype possibility for a normal running mouse. Waltzing mice have to have the genotypes lowercase r, because that is the recessive trait. If you would like to see the results of the mutation for waltzing mouse syndrome, be sure to click the link in the slide. But for now, let's read on to get to the information for the second trait in this problem. Black fur is dominant over brown fur. I'm going to underline this information. Then write out the phenotypes and the genotypes for this trait. For black fur, I'm going to use the genotypes capital B, or capital B, lowercase b. And for brown fur, I'm going to use the genotype lowercase b, because brown fur is the recessive trait. Now that I've listed both the phenotypes and the genotypes for both traits in this problem, I'm going to move on to step 2. Step 2, write out the parent genotypes from the problem. The problem states if a lab crossed one mouse, underline that is heterozygous for both traits with a mouse that is homozygous dominant for normal running and has brown fur, what would be the phenotypic results of the offspring be? Well, based on this information, I know that one parent is heterozygous for both traits. Therefore, I would use capital R, lowercase r, capital B, lowercase b. I also know that the other parent is homozygous for normal running and it has brown fur. So I would use capital R, and lowercase b for the other parent. And I'm done with step 2. Step 3, set up and complete the Punnett square. For this, you need to draw a largish box that will be divided into 16 total boxes. The easiest way to do this is to divide the box in half, then draw lines in the middle of each one of those halves. Do this horizontally as well. Divide the box in half horizontally, then draw a line in the middle of each half. At the top of each box, you will be writing down the possible gametes for parent 1, and on the left side of the boxes, you will do the same thing for parent 2. Keep in mind that you do not donate 100% of your genes to your offspring. You donate half. So parent 1 will donate 1 of the 2 R's and 1 of the 2 B's to their offspring. We have to create gametes for all possible combinations for each parent, and we will do this using the FOIL method. So FOIL stands for first, outer, inner, and last. Like so. The first R combined with this B or the first B to make the first gamete combination. Capital R, capital B. The first R can also combine with the second or outer B to make this gamete combination. Capital R, lowercase b. The inner lowercase r can combine with the inner capital B. That will give you lowercase r, capital B, that we will place above the third box. And finally, the lowercase r can combine with the last lowercase b to give you lowercase r, lowercase b, which we put above the last box. Now, we need to repeat this process for the other parent as well. This capital R can combine with this lowercase b, giving you capital R, lowercase b. This capital R can also combine with this other lowercase b, giving you capital R, lowercase b. This second capital R can combine with this lowercase b, giving you capital R, lowercase b. That goes here next to the third box. This second capital R can also and finally combine with the last lowercase b, giving you the last gamete combination for parent 2, capital R, lowercase b. Now, when we fill in each box, you need to be consistent. Since your parents' genotypes have the R's first and the B's second, keep the R's first and the capital R in front, and keep the B's second with the capital B in front, okay? Now, in order to complete the following boxes, what we do is these letters come down and these letters go over, giving you capital R, capital B, lowercase b. Notice how I organized it. So the capital B is in front and the lowercase b is second. Now keep going. This combination comes down, this combination comes over, giving you this. This combination comes down, This combination comes over. This comes down. This comes over. Okay, now pause the video, complete the remaining squares, then compare your answers with mine. Okay, how did you do? Now, if you do not know or understand how I got the letter combinations for each one of these boxes, you need to stop the video and ask questions. Otherwise, let's go ahead and complete the last and final step. Step 4, analyzing the results. When analyzing the phenotypes for each one of these potential offspring, go through each box and write down the phenotype and make tally marks for each phenotype combination. Now, if you think this is taking a long time, then imagine how long it will take to complete if you rush and make a mistake, and you will have to go back and do it again. I learned this the hard way when I screwed it up in college. So, do this carefully and make sure to take your time. Now, the mouse in the first box is a normal running mouse with black fur. So the rest of these boxes in the first column are the same. Normal running, black fur. So let's go ahead and tally them up and calculate the ratios for all possible phenotypic combinations. To do that, let's go to the next column. Next column. This is a normal running mouse with brown fur. The rest of the boxes for the second column are the same. Let's go ahead and tally them up. Now let's do this for the last two columns. Normal running black fur, normal running black fur, normal running black fur, normal running black fur. Last column. 4 normal running brown fur. So, if you add up all of the tally marks, you will get 8 out of 16 for the ratio for normal running mice with black fur, and you will also get 8 out of 16 for the next phenotype combination, normal running mice with brown fur. And 8 plus 8 totals 16, so you know that you did it for every single box. You have 16 total boxes. Phew. Now, due to the genotypes of these particular parents, we did not have the other two phenotype possibility. Waltzing mice with black fur, and we also didn't get waltzing mice with brown fur. But be sure to pay attention for all of the phenotype possibilities when you complete your practice problems. And do this slowly, consistently, and meticulously, because you don't want to do it again. All right, I hope this helped. Review dihybrid crosses.